allow me to present the okay, could you see it? Yes. A feature of destabilization of AD2 miniature 3 mini-satellite repeat in RHO plus and RHO 0 strains of Saccharomyces cerevisiae. A satellite sequence instability is a well-known phenomenon in a number of human diseases. First of all, this was shown for microsatellite sequences, primarily such three nucleotide repeats, and the factor leading to their destabilization are currently well studied. There have also been reported cases of altered mini-satellite alleles for solved human disease, including various types of cancer. Tumor could appear as a result of genomic instability in a quiescent cell, resulting in an uncontrolled cell division. The exit of a cell from stationary phase of growth to the phase of active division as a result of mutation could lead to the formation of adaptive regrowth. So adaptive regrowth origin and development are used to model some aspect of tumor genesis. Although we know that cancer cells are characterized by significant damage to mitochondrial DNA, which lead to disruption of the function of mitochondria. This aspect of cancer cell is being studied using yeast-split mutant that remain viable and capable of reproducing despite major damage to mitochondrial DNA after their complete loss. So we could see the Rho0 strain. Early American researcher developed a colony color segregation assay for registration mini-satellite repeat alteration in yeast. Insertion of the repeat, the ADE2 miniature 3 allele into ADE2 gene sequence lead to red colony formation due to disruption of various stages of adenine synthesis. Loss of repeat unit or gain of two repeat units within the mini-satellite restores the ADE2 gene function. When such events occurs in dividing cell during the growth of the colony, a white sector appears. Such sensor is stationary phase cell lead to the formation of white adaptive regrowth on red colony surface. This test system allows researchers to study the effect of gene on the stability of mini-satellite sequencer, for example, for gene of the test system or for gene involved in zinc metabolism, as delta Z1 gene. But the influence of mitochondrial dysfunction in this system has not been studied. So the purpose of our research was to compare the feature of destabilization of ADE2 mini-3 mini-satellite repeat and RHO plus and RHO 0 strains of Saccharomyces cerevisiae. The work was carried out as follows. Static mutant isolation was conducted by bromide ethidium treatment of DTK 271 cell was received DTK 271 petite mutant. Destabilization of added 3 minutes, which lead to the restoration of functions of the AD2 gene registered by the formation of white sector and adaptive regrowth of the parental red colony. Since a petite mutant has very long term of receiving of red pigment, we have to use long term of cultivation from 10 to 20 or even 40 days. After this cultivation, we make PCR analysis of cell from adaptive regrowth and other part of colony. We control the change in ecology morphology, and we make the assessment on evasion in the nutrient medium. It was shown this destabilization of added 3 miniature 3 repeat repeat on DTK 271 strain lead to the formation of white sectors on the colony and white microcolony on the red colony surface, which was previously described. Loss of one microsatellite repeat for white sectors and secondary growth was confirmed for DTK 271 strain. PCR analysis for the red colony showed the main band and two additional bands, one of gain of two repeats and one weak band for loss of one repeat. The formation of such additional bands indicates significant stability of the AD2 miniature 3 repeat during cultivation of the colony. During long-term cultivation, 60-day, white microcolony was not formed on the surface of the light red colony of the petit mutant. The appearance of insignificant white sector was reported. Loss of one repeat was also confirmed by PCR analysis for the white sector formed on the colony of the white petit. The apparent result of the absence of adaptive regrowth of the petit unit of the DTK 271 strain are consistent with the literature data on the role of active radical both in destabilization of mini-satellite repeat due to formation of single-stranded bricks in DNA and in the formation of adaptive regrowth during aging of yeast culture. We have repeatedly observed the same effect of petit mutation on a number of laboratory and wild yeast strains. Since we previously found that the cells of original strain are capable of invasion in the nutrient medium after long-term cultivation, we washed off the surface part of colony from the medium and compared the ingrown part of colony of the original strain and its petit mutant. It was shown that the synthesis of adenine is restored is restored as a result of a change in the site of mini-satellite sequence. It led to an increase in the invasive ability in the original strain. For the petit mutant, no such feature in cell invasion was revealed. The data obtained correspond to the literature data on the effect of adenine synthesis on the invasive ability of a cell in both Baker yeast and Candida albicans. We could see on literature data that other mutation lead to inhibition on invasive growth. And you could see how promoted invasive growth for AD plus part of DTK 271 colony. We can summarize our result as follows. No destabilization of ID2 mini-satellite repeat during stationary phase of growth was registered during long-term cultivation of the RHO DTK 271 petit strain of Saccharomyces cerevisiae. Recovery in AD2 function due to one repeat loss during metaphytic division has no significant influence on the level of the RHO DTK 271 cell invasive growth. AD2 miniature 3 destabilization during both metatic division and stationary phase of growth in RHO DTK 271 petit strain were accompanied by sharp DTK strain were accompanied by sharp change in the level of its cell invasion into the nutrient medium. We mean parental strain here. I'd like to express my gratitude to my co-worker who took part in the implementation of this work and the discussion of the result. I'm very grateful to Dr. David Kirkpatrick, who kindly provided us with mini-satellite instability test for this work. And that's all.
Yes, thank you so much, Olga, for this presentation, for this fascinating talk. I remind to our participants, don't forget to send questions in the chat. And just now, at the moment, I have to you two questions. First of all, just can you explain one more time what ADP and RHO gene are responsible for? ADP gene and RHO gene are responsible for functions.
Thank you. ADE gene result for late etap of adenine synthesis. And it is the feature of some yeast that disruption of this gene lead to accumulation of red pigment. This is why it is well-known test used by many yeast researchers. RH gene, this is this mutation because of some disruption in mitochondrial DNA. For example, RHO mutation is a complete loss of mitochondrial DNA. And we are lucky as yeast researcher because yeast could propagate totally without mitochondrial DNA. It's very
Okay, thank you so much, Olga.